안녕하세요.
So we go to quiz first. You should not forget the pharmacokinetic parameter yet. Pharmacokinetic parameter and pharmacodynamic parameter we have. So efficacy, potency is a good side of pharmacodynamic parameter. Toxicity is a negative side of pharmacodynamic parameter. And excretion is one of those ADME. This is pharmacokinetic parameter. Pharmacokinetic parameter is how our body is handling the drugs. Pharmacodynamic is how the drug affect our body. Right? So this is today we are going to learn about very famous the rule, Lipinski rule. So this is kind of a basis of that Lipinski rule. Hey guys, what's wrong? I just explained. What is wrong for ADME? ADME is the collection of a pharmacokinetic parameter, PK parameter. PD should be efficacy, potency, toxicity. Those kind of thing is a PD parameter. PK parameter is, in another word, that is how our body treat the drug. ADME is absorption, distribution, metabolism, and excretion. And this all together, this PK parameter decide how much of those material is available in our body. That's what we call bioavailability. These are all about the ADME. PD is opposite. Okay, not bad. So here the P is a partition coefficient. Partition. What is the C? How about here? C. What does the C stand for? It's not log P. It's a C log P. Small c. Any idea? Nobody? Log P can be measured by experiment. But measuring all those the P value, sometimes it's too much work. So this C is calculated log P. Okay? By looking at the molecular structure, is calculated. That's a C log P. That's a theoretical estimated number. Okay. Okay, good. So this log here, this log is a log 10. So that's why log P is 1. Then upper side is organic solvent, and lower side is the aqueous, the water. So 1 means 10 to the 1. Okay. 1 to 1 is C log P value is 0. If it is a minus value, then the compound goes to more to water than organic solvent. Okay. So that means if the C log P value is higher, is that hydrophobic, or hydrophilic?
Hydrophobic.
It's hydrophobic. Okay. The higher number is the more hydrophobic. That's log P. C log P 1, C log P is 1. That means 10 times more amount into the organic solvent, and only 10% goes to the aqueous media. Sounds like it's hydrophobic, but it's not hydrophobic enough. Most of those drug compounds or organic molecules, they have a higher log P values. And judging from the Lipinski rule, 1 is not good enough. 1 is still too hydrophilic. If the compound is too hydrophilic, it cannot penetrate the lipid bilayer. So it cannot penetrate. And nobody got the right answer. Okay. There was the proper range for proper drug property for log P values. Anybody knows or remember?
5, smaller than 5.
Less than 5 and less than 5. This is less than 5. So maybe okay, you think. But I said that this is too hydrophilic. There's another number. Yeah. Okay. Keep curious and we'll handle it later. Okay. Perfectly wrong answer. Hey guys, do you remember the morphine and heroin? What is the difference? Which one is a stronger drug, morphine or heroin?
Heroin.
Heroin, because? Say again?
Hydrophobic residue.
Less hydrophilic.
Which one is less hydrophilic?
Heroin.
Heroin. How do they make heroin is less hydrophilic?
It changes the OH to something like carboxylic.
They blocked the OH hydroxyl group of morphine by adding acetyl group. They removed the hydroxyl group and they added 2 acetyl group. and then it blocked the hydrophilic site. That's how they made this heroin more hydrophobic. This becomes hydrophobic. That's why their absorption into the body was increased dramatically. Exactly the same problem we had, Valenza versus Tamiflu. Tamiflu, it can be used as an oral drug, but Valenza cannot. Valenza could be used for what form? Do you remember?
Inhale.
It's inhale spray. Okay, because Valenza was too hydrophilic. That's why this compound was not cell penetratable. That's why this compound was only could be used for spray, directly applied to the acting site. Tamiflu could be used for oral drugs. So I expected maybe either the answer would be heroin or the Tamiflu, but Tamiflu still has some carbohydrate-like motif, like hydroxy group. Heroin is fully protected. That's why heroin was most hydrophobic compound. Okay, so it seems like there are many things we need to learn today. So that's the topic we are going to study for judgment of whether one compound is a drug-like or not. Okay, do you see this screen, small molecule drug?
Yes.
Professor.
Okay, good. So for small molecule drug, that is something we are going to study today. Now, when we make drug compounds, we just learned briefly those are the approach by selecting the target, deciding disease, and then do the screening and testing the compound or something like that. But we didn't have a chance to look at those details of the structure of the compounds. So now, if you decide your disease, let's just say coronavirus, and then you select your target, maybe one of those enzyme or maybe binding site on the virus surface, then you can get maybe candidate drug compounds in the beginning. Now, you have compound candidate in hands. You have a target. The target you can isolate or express, you can test it. But when you modify those compound structure, you do not know how they interact in the beginning, the drug and the target. So that's why in the beginning, your approach is you can modify your compound structure, drug structure. It's difficult to modify your target structure because that's natural structure from virus, so we cannot change it. But our option is we can change our compound structure and then compare those activity by changing the structure of the compound, and then you check that activity, whether that change increase or decrease of that activity. So this approach is called SAR, structure activity relationships. Okay. So if you do this SAR, then you can improve the potency of the drug or maybe efficacy of the drug and also optimize their physical properties. So through SAR, if you focus on this potency or the efficacy, you are improving the pharmacodynamic properties. But at the same time, by changing this, you can also change those physical property too. But if your screening is for against isolated enzyme or isolated receptor from the virus, then the compound accessibility to the target site is not a problem because you mix directly the drug with the enzyme, or you mix directly the receptor with the drug, and there is no problem of access of the compound to the site. Okay. In the case, you are looking at only if this is your molecule, and by changing those molecular structure, and you can assume or you can build the arbitrary or the artificial structure of your receptors. So you can assume that, okay, if there is a receptor binding here, maybe this site is a preferred hydrophobic binding, and there is some hydrogen bonding is preferred here, and some charges is preferred here. Then you can assume the binding site of your target protein may have a negative charge here. If there is the hydrogen bonding donor here, then there may be hydrogen bonding acceptor here. Hydrophobic motif, maybe the other partner is also hydrophobic. Charge transfer, maybe there may be some charge acceptor here. Hydrophobic, maybe again hydrophobic here. Dipole, another dipole may be here. Kind of artificial model of your target receptor or enzyme you can draw. Okay, so if you improve by changing, okay, 1, 2, 3, 4 carbon, how about we cut here, make it 2? Then that's better or worse, then you can decide the optimum size here. 1 phenyl or the 2 phenyl or this side, you can try to change, and then you optimize the compound structure. In this case, this SAR, this structure activity relationship, this is to improve PD parameter or PK parameter.
PD.
This is a PD parameter improvement. Okay, you know your target, and then increase those binding affinity to the target. Increase the binding affinity, most likely it will increase the activity. Okay, LD50 value will be down, IC50 may be down. Okay, you improve PD property out of here. This is the main purpose of SAR. Okay, so now let's summarize. Now you have a disease. We wanted to treat coronavirus, and you figure out how many proteins coronavirus has, and then you select which protein can be potential target for the treatment for the coronavirus, but you don't know the exact structure. But sometimes, if you can get X-ray crystal structure of your target, then it's possible you can see those target structure. Now, you have a candidate molecules. If you don't have crystal structure, you can try this kind of SAR and improve your molecular structures and find out best compound from your optimizations. This is what we call optimization. If you have a crystal structure of your target, the next step, the possible other approach is you can mix together this drug with the crystal structure of a protein. Then there is a good chance that this compound can bind to the binding site of your target protein, and it can be co-crystallized. If you can get that much of information, then you can look at how this compound is binding to the target protein in a molecular level of resolution. So that's how we can use our the light accelerator. Okay, then they can generate that kind of a structure, crystal structure, very quickly. Then by looking at those binding mode, we can design further how to further improve of those binding ability. Okay, so through this we can do, if you can do the later stage of structure-based analysis, that we can call this a rational design of a drug development, because we can look at those binding motifs. By the way, all of those the improvement is for PD improvement, activity improvement, efficacy or potency improvement. But there are many reasons why the drugs fails in a later stage, either clinical 2 or clinical 3. And one of those very big problem here is about 40%, almost half of the situations, the problem coming from ADME. Okay, even though you improve those PD property a lot, still there is a good chance of ADME problem. So you have active compound. So that's very good, important. It's high potency, high efficacy, but whether the compound, the drug can reach to the action site or not. If it cannot reach to the action site, it cannot meet coronavirus, then there's no use of this compound. Okay, so this is the PK parameter, and this is, in other terms, how do we measure that kind of a PK parameter? In case of a SAR study, at least you have an input of a chemical structure, output as biological activity. But if you just want to measure those PK parameter, whether it can go to the target or not, how can you measure? That's the problem. Okay? Sometimes even you do not know where is the target. Then at least you can, by looking at the structure, let's just say 2 compounds here. Which compound may be better compound or drug-like compounds? That means which compound can go inside the body. Okay. In this case, we don't have any information about the target. So that's why we cannot judge which compound is better or not. To do that, you need to have a target and you need to have SAR data. Okay. But in this case, if I just ask you, which one looks like a better drug? Then in this case, the question is pharmacokinetic parameters. So pharmacokinetic means if the compound goes into our body, there are several different ways. By injection, or maybe by taking the oral drug and goes through here, and then whether it can reach the binding site or the inside our body. So the other parameter is, one is a pharmacokinetic parameter and defined by ADME. And then the other parameter is activity. So this is 2 basic parameters of the drugs, PK and PD. So PK, if you do not know exactly where is the target, then the first question we ask is whether the compound can go inside our body. Okay, we discussed several times of inside and outside of our body, but simply speaking, the real inside is if you have a cell, and the cell is isolated by this lipid bilayer, and if this is a lipid bilayer, then the compound should reach inside this cell when we edit the compound from outside. Okay, then the question is whether the compound can penetrate this lipid bilayer or not. Lipid bilayer, the outside charges, do you remember what are the charges of outside here? Is it positive or negative?
Positive.
Was it positive? What's the component of phospholipid? You can look up your notebook later. Most of this phospholipid, at best, they are neutral because it's a phosphate there. Phosphate is a negatively charged. If there is any positive charged material together like a choline, phosphatide choline is a negative, positive, altogether neutral. Majority of those phospholipid, because of that strong phosphate group, almost every phospholipids, they are negatively charged. Okay, so there is the difference between positive potential outside, negative potential inside. Okay, that may confuse you. So if you measure those relative potential, outside is slightly positive, inside is slightly negative, but that is a relative potential. That means inside is more negative and outside is less negative. Okay, that's why potential-wise outside is slightly positive, but that does not mean that the actual charge is positive. Okay, actual charge is both sides is still negative. Now, if your molecule outside here, if this molecule is too hydrophilic, then do you remember the thickness of a membrane? One time we calculated those membrane thickness.
4
nanometer.
Again?
4 nanometer.
About the 4 nanometer. Okay, so one carbon-carbon length is approximately 1 angstrom. That's about the 0.1 nanometer. So one layer here is about 20 carbons, and 2 layers is 40 carbons. It's about the 4 nanometer. If you look at compound structure is much smaller than thickness of this cell wall. So that means if the compound is very hydrophilic, it cannot penetrate this membrane. It may just stay outside. If the molecule is negatively charged, then there may be electric repulsion here too, even worse. If it's positively charged, then at least they can reach to the surface and they can bind to the surface. But it cannot penetrate this membrane. Now, opposite case, if the compound is very, very hydrophobic, then what happens, do you think? Then they bind to this surface, and then whatever the membrane they meet, they just stuck to the first membrane, and they do not go in, they do not come out. They are just staying to the first one here. So that means some drugs, if you take in the pill is broken, and then if you taste it, and the drug taste does not go away, it just stuck on your tongue. Sometimes it happens. That means the drug is very hydrophobic. It's almost like a oil structure. Then they are too hydrophobic. Whatever the membrane structure they meet, they just stop on the first one. So then if you inject it, then you inject those drugs through the syringe. Then the first area of injection, the compounds are just stuck there. They don't move in. So that means there is some balance. The drug should be reasonably hydrophobic, but it should not be too hydrophobic. At the same time, too hydrophilic should not be. So our body, so this is perfect meaning of inside and outside if we are only one cell life. But actually, we are much more complicated, and also we have many, many cells in our body. So that's why similar concept we use, bioavailability. If you take the drugs, whether those drugs can be available inside our body. Okay, that inside, for conveniency, we said it's an inside blood vessel. Okay, bloodstream, if we have the compound, then at least the compound can be delivered to every site of our body. That's kind of a convenient surrogate. That's not really the inside our body we discussed. Real inside is inside the cell, right? But still, this is a convenient surrogate, especially if the drug is orally taken, like a pill, not injection. Oral drug is much more convenient than injection. So that's why, if possible, we want to make every drug orally available. So oral availability is a little more difficult to achieve because the drug should go into the stomach. Stomach is a strongly acidic environment, and the drug should survive that acidic environment. And then they should be absorbed through gut, and then they should be entered to the bloodstream. So this is a little more difficult, but this is kind of the optimum ideal cases. So altogether, this parameters is summarized as ADME. ADME again, absorption, and then distribution, if it is taken by orally, and then absorbed through gut, and then through liver, and then enter into the bloodstream. So they should survive all of this the process. Also, they should survive this metabolism. The drug is metabolized by our body, and then eventually they can be removed from our body. This whole process is called as ADME, and the physical measurement of this was suggested by this medicinal chemist named as Lipinski. Sounds like a Russian name, right? Yes, it is a Russian name. So Lipinski, he studied thousands of drugs marketed, and then people has used for many years, and then tried to look at those molecules already commercially the available drugs the property, especially focusing on absorption. Absorption is the first step of ADME. If they cannot be absorbed properly, then there is no need to discuss about the distribution, metabolism, or excretion. So this absorption is directly related to penetrating the cell membrane. Okay. So whether this drug can be good observer or bad observer, that's what he tried to figure out. He looked at in a negative side. In this condition, the drug's absorption is bad. When the molecular weight is bigger than 500. So this is kind of a standard of criteria of a small molecule. Usually we divide the drugs as a small molecule and macromolecule or protein drugs. That critical criteria is molecular weight as of 500. If it's a 500 or less, the absorption is easier. If it's bigger than 500, absorption becomes poor. This is one rule we found. Second one is the C log P. C log P value-wise, if it's higher than 5, too hydrophobic, then it binds to any membrane structure. Less than 2, too hydrophilic. Okay, so that means ideal drug should be higher than 2, smaller than 5. That's kind of optimum conditions defined by Lipinski. Okay, and he also the suggested hydrogen bonding number, if it's bigger than 5, so it's bad. You can think this one is related to this number. Hydrogen bonding is more similar to, hydrogen bonding is similar to water-like property. That means that is more related to hydrophilicity. Higher number of hydrogen bonding means they have more hydrophilic properties, right? And when he counted hydrogen bonding accepted, if it's bigger than 10, that's bad. Then to be good, we just take everything opposite. To be good drug, or to be good absorption drug, or even we don't need to think about the drug. Any molecules to be good absorber, molecular weight should be less than 500. Log P value wise less than 5, bigger than 2. Hydrogen bonding donor less than 5. Hydrogen bonding acceptor less than 10. So many of those numbers are containing 5, 5, 5, 5 by 2. So sometimes this rule is called as the rule of 5. Maybe the exact number was a little off, but to make it easy to memorize, so he suggested this value. And then many papers cited this work. And later, a little original paper was published in 1997. And then this paper was cited more than 10 times, 10,000 times by 2016. And then there was a little more modification was made. And one more rule. If rotational bond is bigger than 10, it's bad. If the molecule is rotatable here and there, molecular structure is not rigid, then the molecule is difficult to penetrate the membrane. Okay. So one more rule here, rotational bond should be less than 10. So this morning I checked again these papers, the citation. Only during those last 4 years, citation was increased almost 6,000 times. So it's even published, and then for the 20 years it was 11,000, and for the last 4 years it's an even higher rate. It was increased. That means this rule is really popular. It's a very convenient rule. By looking at those structure that we can figure out. Okay. So let's come back to this other molecule. Now, let me ask you, this molecule is a drug-like or not? You just learned about the Lipinski rule. Now, the first one was about the molecular weight, right? How do you know the molecular weight? You can count those atom numbers and then calculate. That's one way. Or you can use software, so-called ChemDraw. Have you used the ChemDraw software? Never used? Okay. So ChemDraw was the software to help you to draw this kind of a chemical structure. Actually, that software invented by one master student in MIT about 30 years ago. And he graduated with a master's degree after he invented this software. And it was optimized further and further. Now it becomes kind of a standard drawing software for chemists. So you can draw this molecule, then by clicking this molecule structure, the software itself can generate this kind of information. It's very convenient. Then the software can tell you molecular formula and two mass data. One is the exact mass and one is the molecular weight. Why they are different? And what is what? Exact mass and molecular weight. Why do we have two molecular weight? Any idea? And you can see molecular weight is a slightly higher number than exact mass.
Isotopes.
Okay, Dongwoo gave the good keyword. He said isotope. Do you clarify a little further?
So it's either having a different structure when it has the same combination of molecules,
Structure you said?
or different molecular weights on a different atom.
Because okay, different structure is a little the off concept, but you can say different composition. So let's just say here, this is a carbon, this is a carbon. So we learned how to read this structure before, right? So this is just a dot, but actually this is a carbon. That means it's the CH3 here, right? Now this carbon, actually it has different isotope. Different isotope means which has the same proton number, but different neutron number, and the atomic mass is different. So carbon standard is 12.0, but there are some carbons are 13 or 14. So then if you inject this molecule into mass spec, then you will see as a mixture of different isotopes. So 12, 13, 14, you will see as a different molecule, but chemically identical. That's why I don't want to say different structure. So chemically they are identical, but they have different weight isotope there. That's why exact mass is most abundant isotopes mass is the exact mass. Molecular weight is the average value of those isotopes. So that's why there's a little slightly usually higher number of isotopes, 13, 14, and hydrogen case 1 or 2, deuterium, tritium. So that's why molecular weight is a slightly higher number than the exact mass. Then if you calculate your chemical yield of your reaction, which number should you use?
Exact mass.
No, you should use molecular weight because your material is always mixture of this different isotope. Exact mass is important when you inject this molecule into mass spec. Then mass spec recognize those different isotope material as a separate different molecules mixture. Okay. Anyway, this information you can get from ChemDraw. How about log P? Very conveniently, log P is also calculated by ChemDraw. That's why this C is ChemDraw calculated C log P. Now, up to here, if you have a ChemDraw software, you can get this information. That means if you take an exam, this much of information will be given to you. Okay. Now, judging from this C log P, this molecule is okay or not?
It's okay.
Because this is bigger than 2 and smaller than 5. So this is in the range. Okay. We said this P is a partition coefficient. And I briefly mentioned that the partition is in between organic solvent and water. Water we know, but organic solvent, it can be different options you can use, right? So which organic solvent has been used, do you know, for this partition coefficient?
Octanol.
Which octanol? Octanol also has a different the isoforms. In this case, it's a normal octanol. It's a linear octanol, and at the end, hydroxyl group there. You could choose, so like just octane versus water. That also can be phase separation. and then you can add the compound, and then you can measure those distribution. But in this case, octanol has a one hydroxyl group at the end. It is hydrophobic enough to make two phase, but there is some miscible the boundary. So anyway, and octanol has been used. Now let's count. So this information could be given to you, but other information you should calculate yourself. Let's check. Hydrogen bonding donor, how many there?
1.
Hydrogen bonding donor, so only one here. Do you count this F or not? When you learn hydrogen bonding in general chemistry, hydrogen bonding can be formed OH, NH, and HF is also very strong hydrogen bonding they make. Then should you count this F as a hydrogen bonding donor or not? Donor maybe not, but here there is one hydrogen here, actually proton H+. Then that's hydrogen bonding donor wise only one. How about hydrogen bonding acceptor? How many hydrogen bonding acceptor here?
7.
7. 1, 2, 3, 4, 5, 6, 7. Okay, now we have this question. This F, should it be counted as a hydrogen bonding acceptor?
Depends.
Depends. Okay, depends on this case. Then which should be the call? We count or not?
Not.
Okay, when the fluoride has a hydrogen bonding, it works only as F minus. F minus can form very strong hydrogen bonding. Once this F is bound to the carbon, then they do not have enough electron left over. That's why in this definition, any fluorine bound to the carbon, then there's no exceptions. Any organic molecules, you don't have a free F minus. F, you don't count. Only you count nitrogen and oxygen. Okay, that's the definition that made by Lipinski. So the hydrogen bonding acceptor-wise, this is 4. How about the rotational bond? Difficult? So let's try it together. If the structure is inside the ring, then we can assume that this is a rigid structure. They don't rotate. Now here, this bond, this is single bond, is rotatable. OH group can be rotatable altogether. 1. How about the O and H? Can you count this as a rotatable bond? Actually, this OH we write, but there is OH connected through bond, right? In the case, H is just one dot. H does not have much volume, so we don't count this as rotational effect. If you rotate here, this OH is moving like this. So there is a very big volume effect here. So we count this as a rotatable bond, but we don't count this. Okay? How about this? There is a CH3 here. Do you count it or not? HHH. If you rotate here, then it looks like Barangabi structure here. So they have volume effect here. You count. How about here? Same thing, right? So then 1, 2, 3, 4, 5, 6. How about the HCF bond? Do you count? C and then H, CF, or maybe same thing, C and H. Here again, at the end, there is only one atom. Even though you rotate it here, you don't feel those effects. Okay, F and H is not much different. So you don't count the CF bond. So let me tell you again, 1, 2, 3, 4, 5, 6. Rotational bond wise, this is 6. So now you got all the informations. C log P value, molecular weight, less than 500, between 2 and 5, less than 5, less than 10, less than 10. This molecule, by Lipinski rule, this molecule is a drug-like. This molecule has a reasonable chance to be observed through cell membrane. Okay, let's move to the next one. Second molecule. Now you see those informations. Molecular weight, okay, less than 500. C log P, how do you judge? This molecule is
Not drug-like.
this molecule is too
Too hydrophilic.
hydrophilic. Hydrogen bonding donor. How many hydrogen bonding donor here?
5.
5. 1, 2, 3, 4, 5. Hydrogen bonding acceptor.
Seven.
7. 1, 2, 3, 4, 5, 6, 7. Good. Rotational bond.
Seven.
7.
Let's count together. 1, 2, 3, 4, 5, 5. No objection. 5. You don't count this, right? This is the one atom here, so no count. 5, 6, 7, 8. Which one did you miss? Let me count again. 1, we don't count this. 1, 2, 3, 4, 5, 6, 7, 8. Okay, rotational bond is 8. Now, as you can see here, so this molecule is not drug-like because of this parameter, and also this one is almost reaching the maximum number. So because of this property, we can judge that this molecule is most likely too hydrophilic. This molecule can be used maybe as a spray, but it cannot be used for oral drug. This is the Lipinski rule. And Lipinski rule is measuring pharmacokinetic parameter. So here is some more of the exercise problem. So you can try to solve this problem. And by counting of this, the 3 parameters, then you can judge. At least by given parameter here. Now this one has a little bigger than 500. And you can measure those parameters and you can figure out. Now usually, the cell membrane is following this Lipinski rule. This part, let me skip. And this is a more exercise problem. Famous drugs here: Taxol, aspirin, Celebrex, Gleevec, Tamiflu, and Viagra. So this is practice problem. Should I give this to you as an assignment? You wanted to solve this problem and check it later? Anyway, you solve this problem yourself. And if you have time later, then maybe we can review this together. Okay? So this is the Lipinski rule, which is quite popular and also judging method of a small molecule, whether they are drug-like molecule or not. Okay. Okay, if we have more time later, then we will come back. So today I asked Sejin Sanbe to present important the example of molecular evolution of phage display. So Sejin Senbei will explain to you about phage display today. If you have more time, then we can come back to this group and then practice together. Okay, Sejin.
Let me start it. Hello, this is Sejin. Today we will talk about phage display. Phage display is a laboratory technique for the study of protein-protein or protein-DNA interaction that uses bacterial phages. Actually, we have studied antibody development in the last class. It is really useful method to find out the interaction between antibody and antigen. We will see more detail later. So the most common bacterial phages using phage display are M13 filamentous and FD filamentous phages. Phage display was first described by George P. Smith in 1985 and improved by Greg Winter later. They were awarded the Nobel Prize for Chemistry in 2018. This is a Nobel Prize technique. So here is a principle of phage display. Because phage display is most widely used for in vitro antibody selection, I will give you explanation with antibody development process together. So first of all, it is required to construct the gene libraries. The mRNAs of variable heavy and variable light chain are extracted from human B cell and then reversely transcribed into cDNA for PCR. The cDNA amplified by PCR is ligated into the phage's coat protein by DNA recombination in order to display peptide on the bacteriophage surface. Actually, here is the explanation. The format of antibodies displayed on phage surface can be either SCFV or FAV lesion. So I mean, it can target the antigens. So once the gene libraries are ready, next is getting a higher affinity against the wide spectrum of targets. I mean antigen. Antibody libraries are typically screened by repetitive cycles to enrich target binding phages. This process is called panning or biopanning. By immobilizing a target antigen on the plate, a phage that display a protein binding to one of those targets will remain while others are removed by washing. Those that remain can be isolated and amplified through E. coli infection because this is bacteriophage. And then the selection and amplification steps can be performed multiple times to isolate higher and higher affinity antibodies against the antigens. So this is very simple principle for phage display. And phage display of antibody libraries has become a powerful method for both studying the immune response as well as a method to rapidly select and evolve human antibodies for therapy. So, antibody libraries displaying millions of different antibodies on phage are often used in the pharmaceutical industry to isolate highly specific therapeutic antibody for development into antibody drugs primarily as anti-cancer or anti-inflammatory therapeutics. One of the most successful was adalimumab, another brand name Humira. Adalimumab, an antibody to TNF-alpha, was the world's first fully human antibody, which achieved annual sales exceeding $6 billion. So, according to one review paper, There are 9 approved therapeutic antibodies on the market so far. Actually, the huge success of Humira is due to the patent protection of intellectual property rights. However, almost all of the key patents regarding phage display have expired. The expiration of these patents will allow more companies to create phage display human antibodies, and the antibodies can be available in a low cost. So this is the end of presentation. Thank you.
Thank you very much, Sejin. The question to Sejin and about this phage display technique.
I have a question for the principle in the current page. There is a half cycle between the total cycle. What is the difference between this half cycle and the normal cycle?
So actually, I can say this number 4 is kind of final step to isolate higher binding affinity phage library. So before isolating the gene from the E. coli, they just repeat these steps, the biopanning, and then amplification in bacteria, and then another biopanning, amplification, biopanning, this kind of step.
Okay, do you have other questions? Sejin, would you take down your file? Okay, if you look at here, so this phage in the beginning, phage carries the DNA information. And then utilizing this DNA information, they make a protein. And then their protein, actually there are 2 kinds of proteins. One protein can be made here, or another protein can be expressed here. So phage has a different numbers of proteins, and usually they utilize the tail, a little bigger protein here, or small protein on the cell surface, on the phage surface. Phage is a virus, and they cannot make very big protein, but they can make antibody-like structure. And antibody is 4 chains, one heavy chain, and 2 light chains here. So 4 chains. To make this part, they utilize this single chain FB strategy. Then they can code this structure as a one peptide and express that DNA information inside here. That DNA can be generated by chemically. So you can synthesize those DNA sequence and then insert that into this phage. Then this single chain FB structure can be expressed here. So this is, in a sense, very similar strategy to make antibody, but artificial antibodies. So that means any kind of a protein or peptide, they can be expressed in phage. If it's a big one, they use this tail protein. If it's a small one, then they use this surface one. And then if you utilize this one for the selection, now this immobilized target, it can be antigen, it can be DNA. So antigen means most likely can be protein, or it can be DNA, or it can be small molecule too. So if you wanted to make a small molecule binding peptide or binding protein, you can immobilize your small molecule on the surface here. Then let them interact. Then when this phage can bind to this surface, then all non-bound one you can wash out. Only bound one can survive here. Now here, there's 2 different steps, 2 different approaches you can do. For this bacteria, for this the virus, you can just add the bacteria here. then bacteria can recognize this and swallow this. And then this phage can be amplified in this bacteria. Then you can go this cycle. Okay. Now in this case, you can try maybe the bound maybe 10%, and then second round another 10%. That means maybe best 1%, and next round maybe 0.1%. You can enrich your stronger binder through this pathways. That's one approach. The other approach can be through this the phage display. You can extract those genes, extract out those DNA, and then you can do maybe PCR, then amplify those DNA. During those PCR, you can change those conditions, and you can intentionally increase the errors or mutations. So increase the point mutation, and this is the originally selected one, now slightly modified and generating a little more diversity around here. Then it can go into phage. That's also another possibility. So this is like similar to when we learned about the antibody. So there was a germline antibody, and then through somatic hypermutations, they introduced a lot of point mutations, right? So similar logic here. Then utilizing this one, intentionally introduces similar to this but slightly modified the sequences can be introduced chemically through PCR, and then it goes into here. So this kind of a PCR is called as an error-prone PCR. Easy error introduced other PCR kind of name. Then that kind of a process can be accelerated like this. And you can generate a lot more diversity, germline, and then somatic mutation, and then the effect. Okay. Any other question for this? As you can see, this technique is very powerful, and then number of the diversity in the beginning. Antibody, the germline antibody number is usually million. But in this phage display, you can introduce a lot higher diversity. You can get even 10 to the 9. That means billions of the starting material you can start. And then you can generate this kind of binder through this technique. So as Sejin said, the original techniques, the patent is already expired. So everybody can use this technique. Now, if you select your own antigen, and if you make your own product, then it's totally yours. So general approach is open, and it's a really powerful technique, so-called molecular evolution. So mimicking antibody generation through this phage display, and then you can get this kind of drugs. What is the target of Humira, Sejin?
TNF-alpha.
Then this the drug can be used for what kind of a disease?
Crohn's disease.
Hmm?
Crohn's disease or concern kind of autoimmune disease.
TNF is a tumor necrosis factor. So if you block this tumor necrosis factor, this is kind of suppressing the inflammations. So this is anti-inflammatory the drug, anti-inflammatory the antibodies. So far we have learned many of the anti-cancer antibodies, but this approach can be used for different approaches. As Sejin mentioned, now the antibodies, their cells, the total amount is higher than small molecules. So it seems like antibody or maybe protein drugs is another, the alternate approaches we can take as new medicines. Any other question for this? Okay, if not, we have a little more time to practice of Lipinski rules. Okay, can we try Taxol? If you can solve the Taxol, then we can solve all the problem, I think. Okay, so for Taxol, molecular weight, huge, 850. Already this is off. This is not small molecule, this is huge molecule. Still, Taxol is a very powerful drug, so people use for this. So that means Lipinski rule is not perfect rule, just to give us a general or the guide. But physical property of a Taxol is very bad. Solubility is very bad, and penetration is also not so good. But even you make the chunk of this Taxol not soluble form, still there is a slowly secreting this molecule and still can be used as a very active other drugs. Let's just start with a simple one first, aspirin. Aspirin, if you look at molecular weight, 180, the size of glucose is very small. And C log P value wise, what should you call? 2 hydrophilic. So this one is also, if you take orally, you can take a lot, but many of them will be just washed out. Maybe uptake is not good enough. Still, this drug, very small molecule, very efficient. So this is still in use. Now let's calculate hydrogen bonding donor. Hydrogen bonding donor, how many?
1.
1. Only one here. Hydrogen bonding acceptor.
4.
Everybody follows?
Yeah.
Hydrogen bonding acceptor 4. 1, 2, 3, 4. Rotatable bond.
5.
Dongwon, how many did you say?
5.
That's a 5. 1, 2, 3, 4, 5. Okay, so let's challenge you with the Taxol. Taxol, hydrogen bonding donor.
4.
Is that 4 or 5?
4.
4.
4.
1, 2, 3, 4, 5. You don't count this?
I think it is separate.
It's bound to this carbon, right? It's just
Yeah.
by chance it looks like a close to this oxygen, but it's not connected to this oxygen. So it's a 4. Hydrogen bonding acceptor.
15.
Doesn't say the 15. Any different opinion? Okay, let's count together. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 1, 2, 3, 4, 5, 15. Now, what's the challenging one? Rotatable bond.
Mm. 22.
22, Thomas said. 25,
20.
Someone? Doyun? You didn't answer at all today. Are you following?
24.
24. Doyun said 24. 22, 25, 24. Hakjun?
24.
24. Someone?
24.
Youngmun? So by democracy, it's a 24. It's a majority opinion. Juan?
21.
21. Decreased. Youngmun? Okay, let's count it together. So starting from here. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24. So answer is a 24. If you counted one more, then you think again. If you counted lower, then maybe you missed these two. Okay, it's a totally 24. Okay, so this one, I'm not sure whether we have another chance to the practice, but definitely I will ask you this question. This is one of those the famous the general rule, and it's very the useful rule. So definitely you will see this problem in the final exam. Okay, if you want, if these things are not clear, then you can ask me, and we can review one more time, maybe sometime next class or maybe next week. Anyway, so this week, so we studied about this Lipinski rule, and then I uploaded some of for the last 3 months of news articles about the coronavirus on the website. You have seen it, right? So you read those articles and think in a bigger picture. What should we study, or what should we prepare for the handling this the coronavirus situations? It can be maybe history, or it can be policy, or it can be new drug discovery approach or prevention method, and any kind of views that we can discuss. So you think about your own view, and then on Thursday, you are going to discuss yourself and then make most important the 7 topics. And then among those 7 topics, you select those topics one by one. And then next week, you are going to present those are the different topics. And hopefully it can be kind of the complete, even though it's not perfect, but good series of discussion about the coronavirus. At least how can we prevent, or how can we prepare for? Can be vaccine or treatment or anything. So by utilizing everything you have learned during this semester, you may suggest, even though it's not really working hypothesis or working plan, at least a good the proposal, and then we can discuss whether that's feasible or not, whether that's a good idea or not. It's a good idea, but there may be some the part we need to fix, kind of a thing we are going to try. Okay, so this is kind of a final kind of assignment, or maybe the project that you are going to prepare. So I give you some time to think about it, and then maybe the presentation material the deadline will be by Sunday night or the Monday morning. Then we'll have those discussions and presentation next week. And for this the Thursday, maybe the whole time may be a little too long. So I will prepare for another small presentations for the Thursday. But anyway, that's kind of a plan for this week and the next. Any questions for the plan or for the material that we covered today?
When is the time for our final exam?
Final exam will be on final exam week, and that means not next week. And the date will be Tuesday of our final week. That is 20.
23rd.
23, right? Yeah, 23. 23 in class time. So you guys are coming to the campus this weekend? Is that right?
Depending on the cases, but I will go to the campus after the final final exam week
is
is
it?
finished. Yes.
Okay. Anyway, the current plan is the final exam will be the in phase, the 23, the third Tuesday of last week. Okay. Okay. Thank you, guys. See you on Thursday.
Thank you, Professor.
Thank you,